While we finish our lunch, I'm just going to invite our current president, Kate Daniels, up here to introduce our guest speaker.
Thank you, Nicole, and thank you all for being here today. I'm very much looking forward to the talk that we're going to have and the presentation that we're going to have from Dr. Jonna Mazet. And also, Krista, remind me how to pronounce your last name, because I always say Krista Hani, and I don't know if that's right. Is it? You're saying it's fine, is it right? Okay, so Krista's here from Monterey County, and she works for the Department of Health, the Health Department, and so I'm excited that she's here as well, because perhaps if there's some questions that are Monterey-centric, you could help us shed some light if you have the answers. So Krista's here as well, and I'm really excited to have her here. For many of you who have been members of the Democratic Women of Monterey County for a long time, Dr. Mazet is actually returning to the club. She came and spoke several years ago, and at that time she spoke to us about her work to save the California sea otter when coastal runoff was carrying infectious pathogens that were killing the otters. So even if you're not one of the million people that like those cute pictures of otters that the aquarium posts and videos, you do understand that what kills animals kills people. And so we're really lucky that we have someone here to share with us, someone who actually drove 4 hours down and is going to drive 4 hours back to share with us her global, comprehensive, and collaborative approach to resolving problems just like this. She's a woman's scientist in the STEM field, where women continue to remain largely underrepresented. I'm sure she experienced all of the usual roadblocks and obstacles as she rose to become a leader in this field. But I also assume that there were many strengths and insights that she brings to the field as a woman that have aided her in the development of this work and led to the successes that she has encountered with her programs and her projects and the initiatives that she's led. She's going to speak to us today about her global work at the interface of animals, people, and the environment to solve complex problems impacting health and conservation. She's a professor of medicine and epidemiology at the University of California, Davis. She's received numerous awards, and many, many, many publications were listed with her bio that I was researching earlier today. She's the executive director of the One Health Institute at UC Davis and the global director of the PREDICT project, which will be the focus of her topic today. So please join me in welcoming Dr. Jonna Mazet.
That was a lovely introduction, and I'm also going to be your AV tech here for just a second. Super happy to be here. Thank you so much for having me back. I guess we were talking about it might have been 10 years since I was originally invited by Chris and John Walton, who are here and are wonderful citizens, as are, I think, all of you being here. And I know it's a very tumultuous but also exciting time to be in this society. And so I'm here to talk to you not so much about politics. I'll try not to. Actually, you don't need to hear from me. I'm not an expert in that. I'm just hopefully an engaged citizen as well. But I admire your mission and all that you're doing. I also just want to thank you all for encouraging Dr. Hani to come. It was such a wonderful surprise. I haven't seen her in a very long time, and she was my first graduate student. So, it was just like, I'm perplexed. So it's a special day, and also maybe she makes me nervous. I haven't talked in front of her. But there's some interesting things here, because Krista, you heard about, when I came before, I was talking more about sea otters, but I was already moving sort of my career into this arena of One Health, and I'm going to talk to you about a little bit more today. And that means that we're looking not just at a single species or single focus, but a more holistic focus, thinking about the planet and all of the populations on the planet as more of our patients. And her career went in that direction too, working for the public health department for the county of Monterey. And so there's just some interesting, just paths that we take as we evolve as women and scientists. So it's wonderful to be with you. So today I am going to talk about the zombie apocalypse. And if you didn't come for that, you can go now. So some of the examples of things that are very worrisome for the general public. So when I started working on emerging infectious diseases and started that from sea otters and coastal pathogen pollution and all of those things, it brought to mind, what is happening that our sea otters are getting a land-based pathogen that are killing them, and it's a pathogen that shouldn't be in the sea, but it also shouldn't be at the proportion, even if it's making it to the sea, it's in a higher proportion than it is on land, and it's a land pathogen. So it just starts to put together all of these questions and a puzzle, and that's really what epidemiologists like to do. We look at patterns, we try to solve puzzles. And so this puzzle that I put in front of you, it looks more like, sort of the press, capitalizing sometimes on our fear, but expressing our reaction to what's going on in society. But is there something to solve here? Is there something we can do to help reduce our risk from epidemics and pandemics? And I'm pleased and honored to work with a consortium of people all around the world that are looking at new ways to approach these things. Because, sort of the Albert Einstein quote that I like is, insanity is doing the same thing over and over again, expecting the same result. We are doing that in so many ways. I don't get me started on the political side, but if we just think about how that plays out more dramatically, as many things feel like they're playing out more dramatically today, if we think about our population growth. So we are exponentially increasing the numbers of us running around on the planet. And when we do that, we are abusing even the resources of the planet and impacting. We need to figure out how to live, and can we figure out how to live sustainably with all of the other populations on the planet that make this planet function. And what we see along with the growth of the population is we see these episodic spillover events that result in actual disease outcomes, epidemics. And we are seeing on the order of 3 new emerging infectious diseases in people every year now. And, sorry, okay, I'm going to try not to be scary, because that one is a little bit scary. It should be for all of us. But, okay. That's not me. I didn't touch it. All right. Okay, so the scientists among you, professional or amateur, will say yes, but we are better at finding them. We do better surveillance. We have better diagnostic tests. That is correct. This 3 per year is an increase adjusted for our ability to find them more easily and our improved health systems and detection. So it's a real increase. And what happens when we do have them? Sometimes we are having spillovers, and I'm going to talk more about what I mean by that, but sometimes we are having these little new emergence of infectious disease and we don't notice it. That's probably most of the time. Sometimes we have them and we don't diagnose it. Do we have any physicians in the room? No, so physicians are our first line of defense of noticing these new emergence of infectious disease, but also emergence in a different way, emergence in a new location or a new demographic group, right? So there are other ways that we can measure emergence, but often we don't have any way to detect or diagnose them. So 70% of fevers in the world go undiagnosed. You just get better or you die. That's just the truth. Also a dirty little secret, they lay their hands on you maybe if you're lucky enough to be able to go see a physician, but often there's no diagnosis. We don't give them the tools to diagnose something that's new because we don't know how to do it. So when a new pathogen appears on the scene that could cause an epidemic, a pandemic, or just you or me to get sick, if you are lucky enough to have good health care, you can go to the physician, you can check the boxes on your diagnostic tests. You've all seen a lab test form, right? Somebody had to check the boxes, electronically or physically, to say what to test you for. If they don't know what you have, there's no box to check, right? So they will check a lot of boxes, hoping one of those things is the thing. But if it's new, there's no box there. So that's a problem. We need to get ahead of that. We need to think about that differently. Because we can't afford this. So when we have things like SARS, more recently MERS, Zika, the numbers for Zika are going to jump there as we keep figuring out how much it's costing us, on the order of about $40 billion every time we have one of these big ones. Okay? If we have a influenza epidemic, okay, not stomach flu. We're trying to learn to retrain ourselves as scientists to say influenza so that generally people will recognize that a lot of the things that we call flu around here have nothing to do with influenza. Okay, influenza is the scary one. Hopefully you're getting vaccinated. Hopefully the vaccines will get better. Hopefully what I'm talking about today will help us make those vaccines better so that you will want to take the time and the energy and all of those things to get vaccinated. Yeah, we just, all right. But if we were to have one of those similar to the 1918, so commemorating kind of 100 years since the horrible 1918 influenza, Spanish flu pandemic, if we were to have that, it would cost the economies of the world about $1 trillion. And if we annualize our costs of that per year, we'll be spending for decades about $570 billion per year to recover, because whole communities, governments, things crash when you have that portion of your population get sick or die. Okay, then you just lose whole communities, and yeah, that's enough. That's enough scary talk. All right, this is part of what's going on. That we're moving around the earth differently. We're using our resources differently. We are noticing now physically in our daily lives, most of us, change, like to our climate and other things. So we are having an influence on the world. It makes it a little bit lighter, right, that it's doing that? It makes the topic a little more it doesn't do it here, so it's fine for me. But we're moving around and we're using the earth differently, and that's putting us at a different risk. So about 10 years ago, probably just after I left here last time, there was a group in our U.S. government through the U.S. Agency for International Development, which is not a usual source for a person like me to approach to get funding or think about doing the kind of research that I and the great team that I work with do. But they said, we want to change this. We think there's enough evidence coming out of teams around the world that it's time that we do something different here. And a motivator for them included that they're the ones that actually buy the vaccine. So when there are these epidemics, epidemics happening around the world, they are the ones that do the right thing on behalf of our government. One of the groups, there's lots of groups that do the right thing, but one of the groups that do go out and try to provide vaccine. Now, the problem is they are often providing it too late for it to be useful. So then it is a gift from the American people. That's their tagline. That's on everything. My own jacket usually included. I have a business card, so but if they're doing that at a time when it cannot contribute to the solution, are we really doing the right thing? If we're just fixing that first scary slide, right? We're just making people feel better, but we're not really fixing anything. So another group of crazy people, these are my people that I get to work with, developed something that USAID named the project the PREDICT project, but we kind of, first we're trying to call it the Smart Surveillance Team. We decided we could work on that really hard challenge, and that is, how do you find and know what's coming next? And can you stop it at its source so that it doesn't get out of control? And I think most people now can sort of think about, conceptualize, because we know so much about the West African Ebola epidemic that devastated communities, as I mentioned, killed about 11,000 people, a little more than 11,000 people, primarily in 3 countries in the West African region. If we could have controlled it in a village, could it have been prevented from spreading and from things like schools in Dallas being closed, right? Huge reactions that we had, airplanes being disrupted, right? These kinds of costs go into all of these responses, and the fear that they generate lasts for some time. They last about until we have another one, like Zika. And then what happens? Those of us working, studying, trying to fix, solve the puzzle, get told, no, I'm sorry, half your money's going to Zika instead. Please study that now. Like, whoa, wait, it's no wonder we never fix this stuff. We're just reactive. We're not proactive. We're not studying it holistically. So this group of collaborators, which includes the ministries of health, agriculture, and the environment in 35 countries. In the first 5 years we were working in Latin America. Things changed, politics, other things, and we continued in Africa and Southeast Asia and South Asia, which have been involved for the whole 10 years. We certainly got moved somewhere in the middle of those 10 years to be working in West Africa, where we weren't before. So politics plays in all of this as well, and so we can't forget that. And we decided to take this One Health approach that I mentioned. And we took the One Health approach because we believed and were committed that working together across disciplines, we could do a better job, that we couldn't in our ivory tower in the university with one sort of disciplinary training fix this problem. We needed to stop doing the same thing over and over again. We needed to join together, and we needed to benefit from each other, learn from each other. We talked a little bit, that's more of a woman's approach a little bit sometimes, if you think about it, to science and problem solving. So many hands make light work, as my mother-in-law would say. Yes, in different contexts, but it applies. So, now, why did they I'm a veterinarian first by training, then an epidemiologist, animal and human. But why did they let a veterinarian working on CRs, for goodness sakes, lead a global consortium to try and save the world? I don't know. But I think it had more to do with this issue, and that is that the majority of these emerging infectious diseases come from animals and our contact with animals, and understanding what's happening in the animals and our human behavior that's driving changes in the animal populations, as well as our interactions with those populations, has the most to do with what's coming next. So they needed some people that knew about conservation and wildlife, because most of those emerging infectious diseases also come from wildlife. Now, here's my little secret. That is not surprising scientifically. If it's emerging, what does it mean? It means we didn't really know about it before. Okay, so if we didn't really know about it before, that's probably because evolutionarily we didn't see it, or if we had seen it before, it hadn't really caused a huge epidemic, right? So that we recognized it. So where are we going to get those new things? We're going to get them by interacting and catching things from other populations. So we become the naive population, right? And so we do that because we go into caves in deep forests to get new minerals to make your cell phones lighter, right? We are changing the way we are interacting with wildlife and all of these other species. What else do we do when we build that mine or a dam, let's say, in South Asia or in, doesn't it bother you? East Africa. Could you just wiggle that gray cord for me, just in case it's that?
Yep, it is.
All right, we'll try it. So when we do that, we do that, depending on where we come from, what cultures, there are some big global players that tend to build those kinds of things, infrastructure, roads, highways, dams, mines. Those folks often do what? They bring their own workers from their countries to that country where they're doing that. They also bring new food preferences. They also bring their domestic animals, mostly for food, and they bring them into areas that haven't seen chickens or goats or pigs, right? And they bring them and let them mix with the monkeys and whatever happens to live there, allowing this perfect mixing vessels for spillovers or exposures to new pathogens. All right, so we have this opportunity for an enhanced flow of pathogens. Also, we're just crushing each other on this earth. There's just not that much more place for us to go. And we share resources, especially water, so we have to think about that. And that brings those health risks, and it also leads to us moving around and changing. And we have war, famine, all those other things. Allows us to move around, or requires us to move around to get into better situations. What does that do? That just keeps this thing going in a vicious cycle. So, I'm going to get sciencey on you here for a second, but this is the epidemiology, the epidemic curve, okay? And so if we're thinking about an epidemic curve, usually we just talk about that red part, and that means as people get infected, you see cases over time. And so if you just bear with me there on the red curve, we want to see that curve not occur, preferably, but we'd also, if it's going to occur, if you're USAID and you're providing vaccine, you want to do it on the timeline closer to me so that that curve goes down. You impact that curve, right? So your end goal is really, if I'm going to buy the vaccine, I want to be making friends around the world with that vaccine, but I also want to do good and see that curve go down. I want to have a positive effect. So if, though, you don't know what's out here in the green, that's all the wildlife stuff, all those viruses. Let's just say, I'm going to stay mostly with viruses here. The viruses that are circulating out there, we don't know which one's next. We don't know which one's going to interact and spill over into domestic animals, that's the blue curve, or humans directly. When it gets into domestic animals, it could amplify, just cook along for some time before it spills over into people. So that's what we think has happened with the Middle Eastern Respiratory Syndrome. Everybody heard of MERS, right? Okay, so people are really worried in Saudi Arabia. We talk about the Hajj and people coming together in large groups, and then we're worried about travel and all of those things. Well, if we have bats, evolutionary host for something like MERS, they start to mix with camels because of where they live, in a barn, right? Anybody have bats in their barn? Bet you do. All right, they mix, don't worry about it. It's fine. We'll dump them soon. We'll just ignore them. They mix, the camels get it. It cooks along maybe for decades before we notice in those, maybe we've been having those fevers, right? But nobody noticed what's happening until enough people get sick, all clustered in time, and maybe even there's some modification of that virus so that it can spread from person to person, that we really start to get worried over here in the red curve. So what our goal is to understand what's out there so that we could start to forecast what might be next, rank the risk of what might be coming, understand the behaviors, all of the locations and the occupational situations that might cause contact out here in the green area before it gets to the blue and red area. But if it does get into the blue area, we want to be able to pick it up So we need surveillance improvements in our domestic animals, especially our food animals, and also laboratory diagnostics that we can pick it up when it actually does happen and get into people, because we need that curve to go down. So this is the sexy kind of environment I mostly spend my time in. And I call this the flu of death. See, I have to say, it doesn't sound as good. Influenza of death doesn't sound as good. But this is what we worry about. We worry about these mixing spots, okay? So chickens, avian influenza, they're actually the most common victim of avian influenza. They get avian influenza. They mostly get it from each other or from wild birds. And in those wild birds, it may be a strain that doesn't make the wild birds sick because it evolved with that flu. So the chickens tend to be ones, if it's highly pathogenic, they start to die, it gives you a big warning. Actually, the world has improved and understood that, and now we have methods in different countries handle it differently. But fortunately or unfortunately, pigs, mammals, but also act a little bit more like us with influenza. So the mixing here is things that can combine with the pigs and the chickens and the things you can't see. The bats, the rodents, the shrews. This is this close to these people's dwelling. Those same individuals, not the pigs or the chickens, but the rodents and the bats are also going in and out of the house, and certainly people are going and getting eggs and meat and feeding these animals and all of those things. So what's super surprising is people are really willing to let you swab all of these animals and take samples from their water and the ground, and if you ask them, they will go in the back room and swab themselves. I never thought they would, but I learned a lot. I've been learning a lot. People want to do the right thing. People are good, and they care not just about their own health, but also the health of their community. This is from an area in Kathmandu where the restaurants buy food. Anybody eaten in a restaurant in Kathmandu? Okay, not scaring you, just cook it. Okay, so this is what we needed to do. We took the best data we could find in the world and we started to make those forecasting models, and really soon what we knew to be true was true. We didn't have enough data to do a good job to create like a weather forecast for emerging infectious diseases. So we identified to the best of our efforts places where we should go that are hot spots, have all the perfect storm kind of situations where we should be looking for those viruses. So we got out in the field. We coordinated with the communities that we worked with so that they became the front line. They had phones they could get with us quickly and say, hey, something's going on here. So they became first line surveillance kind of officers out in their communities. And we got that data back into the model, but we also got the samples into the lab. We still had a challenge. Anybody follow me? What's the challenge?
Funding?
Money's always the challenge. Thank you. They actually funded this quite well for this kind of study, which was surprising, given climates and things. You still don't know what to test for, right? You still don't know how to find it. You don't know what to do. And now where are you? You are in the most resource-restricted places in the world, so they don't necessarily have the great labs that we even have here, which, oh, by the way, aren't going to diagnose you for these things if you go to the hospital, unless there's a couple hospitals in San Francisco, one in LA, and maybe two in New York. Okay, no scaring. So we wanted to look more closely and figure this out. So we had to come up with a platform to do this testing in the most resource-restricted places in the world. The great news about that is that means your hospitals in small towns even can do that, right? So we were learning with our colleagues in these countries how to make things happen. Amazing things happen. So it's been so enriching to work together as teams from brilliant people all over the world, but including in these resource-limited countries. They're so scrappy. I feel like also the wildlife people, we never could get funding, right, to do wildlife studies. And we were scrappy, like we just figured it out. You just figure it out. And the same thing with the laboratory platform. We said there must be a way. We know, some of you may have heard about metagenomics, and there are new black box technologies where you can find, it's going to be on your kitchen counter soon. You sit and you say, oh, that's what I have. Okay, that isn't quite ready for prime time, but when we started, and even now, it's quite expensive. Not so much, it's gotten cheaper and cheaper and cheaper, but for the actual technology, but because you need bioinformaticians, you need these brilliant people sitting somewhere else analyzing the data that's coming out of that, and those people are few and far between, which means supply and demand, but very expensive, right? So we needed to keep figuring this out. We couldn't use that metagenomic approach, too expensive, not applicable. If we leave, the countries won't be able to do it, and that's not what we started to do this for. We started to do it for sustainable capacity building. So we kept looking, kept figuring out ways, and we started to look at the interfaces where we thought were the hottest spots, the hottest interfaces, meaning linkages, really ways that people are interacting with the species, that our models, to the extent we could do a good job, were telling us we're the highest risk species. So monkeys, not too surprising, they are very closely related to us, so we share more easily a lot of viruses. So they can get things we can get things they get. We sometimes get different diseases when we share them. So our herpes virus that just causes a cold sore for us can kill them, theirs can kill us. But we can share them, we can both get them. So there's things like that. So we know that from history, again, going through the literature, all of that. We know that, so they're high on the list to start. Bats, we're seeing these SARS, MERS, all of these things that are worrying us, so bats high on the list. This is a bat condo. If you build it, they will come. So we didn't build this for our purposes. We can capture the bats without, causing huge behavioral changes in them. But this is a huge developing industry in Southeast Asia, but building these attractants for bats and harvesting the guano. What do they do with the guano? Yeah, so it's going straight on fields, which is going where? Yeah. Okay, so maybe if you put it out in the sun, in UV radiation, you will kill all the viruses. Maybe we should find that out, right? We should find that out. We shouldn't tell them, stop this thing that just took your family from sort of the lowest rank of poverty in the community to some middle income and say, oh, but you should stop that. That's bad for the community. That doesn't work anyway. Nobody will do it. We should say, does this work, and how can you protect yourself when you're over there, because you're the ones cleaning that up and putting it over in the sun. And if this works, okay, it works. It's not the highest risk thing. If it doesn't work, then we got to figure something else out. We've got to work together and figure out another way to live and have economies. This picture is from a downtown big city. Okay? Most of the people in this, well, I'll just stay with Kathmandu because I already blasphemed them. But in Kathmandu, there are trees, this one is not, but there are trees like this full of bats, and people walk under them, going to work all hours when they're getting up and really active, so they're urinating, defecating on the streets as people are walking to work. And we asked, when we got there, where are their bats? Like, we obviously knew this big thoroughfare, which is right by the old palace. Where are the bats? We don't have bats.
What?
This is another interesting thing. People don't notice bats. They're noisy, and they're like, oh, the birds that sometimes fly at night, those are the ones. So it's just an interesting issue. We don't actually really like bats, and there are lots of evolutionary probably reasons for that, and so we ignore them, pretend that they're not there. Unless you like to eat them. And then you can buy them in the market. Now, I did mention cooking things. These are not cooked. They look like it because they use a blowtorch to take the fur off to sell them, because nobody wants a furry meal. But they're still raw on the inside, so they still have viable virus available. But that's another thing we needed to check. If the virus wasn't viable in them, then we shouldn't be telling people, just because of our own preferences, that that's a dangerous thing to do. Don't put those bats in the food chain. So we need to do a really good job as scientists and as the global community working together to work off of evidence, right? Evidence-based recommendations and decisions. Otherwise, we're no better than any other nonsense, just pointing fingers and blaming. So, okay, I said I wouldn't do it, I'm trying not to.
It's okay.
All right. We also developed techniques. This is not what it looks like. This is, yeah, it's a group of women, so that one's hard not to say. This is actually a rope technique. Some of the places where we go, the monkeys are revered or worshipped even, or protected because they're endangered. So we had to figure out new techniques to be able to sample them without capturing them or disturbing them because of their protected status. So we worked with zoos and primate centers and came up with techniques that we could get this, anesthetize a monkey, swab the inside of its cheek, and we could get the same result as if we threw a strawberry jam covered special rope to the monkeys and have them chew on it. And would they give it up? Yes, they would. They'll spit it out. They'll chew on it for a while, and then they say, this is not something to swallow, just like you would, and they'll spit it out, and we can get it back. So, but the kind of rope mattered. We would get different success off of different kind of ropes. The strawberry jam versus banana versus peanut butter versus anything else. You could, all these things matter. So it's a very interesting long haul to get to the place where we can start to do this. All right, which is really what I should be talking about as a scientist. So I've talked about this. We work on the wildlife looking for the viruses in that, we call it zoonotic pool. So zoonotic meaning going back and forth between people and wildlife. So we look for that, in that zoonotic pool, what viruses might be there. I mentioned rodents, but didn't say. Those also live and adapt easily to people, so they're high on the list. Bats, similarly, we're finding the most new viruses, probably because we don't like bats, so we didn't study them earlier, but the most new viruses in bats. They're also a huge diverse order within the mammals and primates. But what else do we need to do? So we can find a lot of viruses that have nothing to do with people. So we need to start looking into people. Those guano farmers, right? Are they getting something? They will say to us, sure, you can go sample, but we're not going to do anything because we're not sick, and we don't get sick, and we're not worried. We've been doing this for 5 years now. Everybody's fine, okay? So we should listen and figure it out. Are they sharing anything? So are they sharing anything with the bats and the feces that we're finding on their farm? If they're sharing something and they're healthy, maybe it's not a pathogen. Maybe we can get it, but it doesn't make us sick. We can get a lot of influenzas that don't make us sick, but every once in a while they change. So they still need to be on the target list for monitoring. But the other thing we did is we started going to all the clinics that fed from those people that were in high-risk situations, those interfaces, those occupations, and starting to look at those fevers that are going undiagnosed and say, what do they have? And are we finding that in the wildlife as well? And so that's giving us clues, right? We're after that puzzle, putting that puzzle together and figuring out if we can do a better job. This really meant that we were also building the plane as we were flying it so we could train people and train ourselves, but also train a workforce of people that were ready to respond to outbreaks, that knew how to safely sample people and animals without hurting themselves or hurting the animals, that knew how to interact with communities, because if you just show up with communities in these suits and say, I'd like to test your meat, you can have some trouble. So there's a huge community engagement portion, and when you're engaging with the community before you're ready to start it, you are learning, and they're telling you, hey, you're doing it wrong, dummy. You should look over there, that's where everybody's getting sick, right? They have this huge level of knowledge, and nobody asked them, so they don't share it, right? So we're learning and learning from the communities. This tells us our One Health workforce that needs to be multidisciplinary needs more sociologists, needs more economists, needs more medical anthropologists, right? So we're starting to also build and bring together all of the disciplines that can help us solve this issue. And we needed to figure out the lab piece. And what we did with the lab piece was we have a real excellent team of virologists that said, well, you know, we could use this technique and just look for sort of the conserved or the basic part of the genetics of the virus that is similar across all the viruses in a viral family. So sort of the progenitor part of their RNA in this case. And then once we find positives, then we can sequence. And so we can even do older style, not the metagenomic style sequencing, but the more traditional style sequencing that costs a lot less. That's because the other one's more fancy. The older style sequencing, Sanger sequencing, we can do for like $6 now. So you can start to find ways, and it's funny, but everybody wants to do the new stuff, supply and demand. So the old stuff, a lot of the labs where we were working in the country said, yeah, we have a conventional PCR machine. That's old technology. No one here knows how to use it, and we don't want to. We want the new stuff. I'm like, okay, but we didn't get full benefit of the old stuff. Can we do that? Because that we can afford, and, oh, by the way, turns out more sensitive. So we identify that progenitor piece, and then we sequence the whole part of the positives, because only a small portion are positive. Otherwise, we'd be having these diseases every day, right? And when we find ones that are related to SARS or MERS or HIV or all of those, then those give us targets, and we start to understand what might be the next one. We looked only in taxonomic viral families that have caused disease in people in the past. So that's about 24 viral families that we were interested in. There's 28, but 24 that were really targets for us. So,
I want to make sure we get to Q&A only because I know there's a lot of questions. So just FYI.
How much do you want?
I just want to be able to get at least 15 minutes of
Okay.
Q&A, and if we can do that.
No problem. And this one, just talking to you about the amazing teams. These are my friends and colleagues at Sokoine University of Agriculture. With the money from USAID, we couldn't build any buildings. We could just put together this surveillance program, right, and start to find the viruses. And they said, well, the only place we have a laboratory where we can even try to do this kind of work is the lab where we train the 18-year-old first-year college students to do microbiology, and they're learning to pipette. I'm like, oh, not the world's most dangerous viruses for the 18-year-olds just coming in. Anybody have 18-year-old children's graduates? Yeah, you don't want them in there. So they took 2 shipping containers, stacked them on top of each other, 40 grand, built the most beautiful viral laboratory, viral discovery laboratory. It mimics all of the functions of my laboratory. 40 grand, mine's probably 7 million for the same thing. So, scrappy. All right, so what happened? Well, this is what people think is sexy. We found over 1,000 viruses, more than 80% of them brand new to our knowledge, right? They're not new, been around for millennia, but those are all new viruses that are in viral families that have caused epidemics, disease, even pandemics in people in the past, and they're now new to us. So we know that they exist, and we now can watch for them, right? And we found a technique that could be done fairly reasonably in these communities. We also found, as we were going, that our technique also picked up about 210 known ones. So some bad actors there, ones that we know cause bad disease, but we could use the same platform inexpensively to test for knowns and novels, right? So that is a big cost savings, a big efficiency, and it has implications for how we do things in just general medicine here. So that's the part people think is cool. I think this part is cool. And that means that there are 4,300 more people now than there were 10 years ago who can help with outbreaks, can help with surveillance, who can help in the laboratories, who can really change the world, right? And can build their careers going forward to keep studying this to really solve the problem. For me, that's what's really happening. And they're doing that in 60 laboratories around the world that had not this capacity 10 years ago. So, so that's really where we are. I could go a lot more into a lot more science and what that means, but what it really means is we have a lot longer to go. We have a great distance to go. This helped us understand how many viruses are probably out there, because now we know if you sample a certain number of animals, individuals in a certain species in a certain location, we know what that curve of discovery looks like and about how much effort you need to put into that. So we know there's probably 500,000 viruses out there that we should know about. Doesn't mean they're going to make us sick or do any of those things, but we should know about them. We also know that we need a system. You see me? That we need a system to rank them so that we can start to target where do we look harder and which ones do we look at first. We can't continually surveil for 500,000 viruses. But when we started, there were less than 400 viruses known to people. Okay? So we've dramatically changed the way we look at these kinds of viruses. And I think that puts us on the path to being able to have the information to do better with vaccines, to do better with human behavior conversations, if not change. Behavior change is hard, but I think we're on the path of doing things differently. So thanks for letting me come talk to you.
I'll let you just.
Okay.
Yes. Jonna, why are bats, and let's say not guinea pigs, for example, why are bats so pathogenic and other critters that people eat around them a lot or not?
Yeah, I'll try to repeat the questions because I know you all can't necessarily hear when those folks that don't have microphones. So the question is, why bats and not guinea pigs or some other species? That's a big topic of debate. There's a whole school of thought that it has something to do with flight, that the metabolistic pieces and evolution that goes along with flight, because remember I mentioned influenza and birds and all that, birds are important too. Less important, probably because I can answer that one more easily. Yes, they fly, so that makes them risky, but they're not mammals, yeah. So they're not as closely related to us to make us as susceptible to their things. Though when they have something we're susceptible to, we really need to worry about it. Guinea pigs, so there's a rodent camp versus a bat camp. And I don't care, I just currently just find all the viruses, but really what it seems to be in some situations is that we've been exposed to rodents for a really long time, we've lived with them urbanly, so it's not that we're not susceptible. In fact, if we used only history to direct us, we would say rodents are really a lot more important, and that's the rodent camp. But we tend to not see as much new stuff from rodents, probably because we've evolved. We keep them as pets, or we eat them, or they're pests, right? So we've just been exposed. Yes, ma'am.
First of all, a wonderful talk, and we really appreciate what you've done. You focused on virus. There is a chronic wasting disease that is traveling through the deer population and now is in 26 states. It's prion-based.
Yes.
Can any of your technologies or techniques that you're using in surveillance be used? I mean, we can't
Sure. So the questions about chronic wasting disease, it's a disease of primarily wild ungulates, so deer. We worry about deer and elk. It's a big problem in the Midwest and now the Rocky Mountain West, and sort of it's been spreading very slowly, but it's spreading slowly because it's a prion disease, and prions grow very slowly, so it takes a long time from exposure to get sick. And so the question is, can we use this same technology? The answer is yes, but it's probably not the most efficient technology for prion disease. So there are other better ways. We can detect, there's been a couple decades of development of diagnostics for prion disease. The real reason why we are doing a huge amount of surveillance around that, it has more to do with it being a wildlife disease. And remember I said not a lot of wildlife on it, right? So that's more of the issue there.
The worry is crossing over to humans, and then we've got
yeah, and so we worry about that. There's some evidence that's not very convincing, right? But there is evidence that we should worry about that. There's also, we know a lot about prions, and we know they can jump. And I will say that we've now come up with 40 factors to help us rank viruses on which ones should be looked at more carefully first, as resources allow. And one of the most important factors is how many species it can infect, and how widely evolutionarily apart those species are, how different those species are. So if it can infect a giraffe and a mouse, we're more worried about it than if it can infect two similar kind of bats, right? So that also holds true for prions that are good. They're slow but good jumpers.
Since people are not willing to like slow their reproduction, it doesn't look like, and it seems
We
to
have.
me that yeah, well, and yeah, in some areas we have. Do you find that the viruses have grown with human population? And number one. Number two, what if we only had, and how it had always been there, like 1/3 of the population we now have?
Mm-hmm.
Where would the virus count be? And if, what's the purpose of anything that exists that's alive is to reproduce,
Okay.
right? Including viruses, right? So if the Earth has any intelligence at all, I mean, that's a joke, but, well, wouldn't you like send the virus to decimate the human population so that everything else can survive better?
Okay, so, and you're taxing my middle-aged memory by giving me 3 things or 4
All that can
at
be
once.
false and not have any reality at all.
Okay, there's an issue where the base issue that we've agreed on already, or I made you agree with me without asking you, is that there's an issue that the population is increasing. So that's absolutely true. What does that mean? Where are the viruses coming from? Is that because we have more humans, or is that because we're interacting with the Earth differently because there are more humans? It's a great question. I study emerging infectious diseases, which allows me to avoid that question because I'm only looking at new things. And new things are generally mean we haven't seen them before, or we haven't seen them in that location before. So things that aren't new, like new influenzas, are not new to me, right? They're influenza A. And so I worry less about those, but those are being impacted by just more humans. All right, but the brand new ones that we haven't seen in our evolutionary history seem to be more impacted by the way we are using the Earth than they are that we just have more humans. But they're both, the amplification and spread of the new things, hugely impacted by that there are more humans and that we run around the Earth very quickly. Me, the worst. I got status on every airline. So I'm going to stop there because there are more questions and your questions are too hard.
So I cannot get political.
Okay,
It's just who
it's
I
your
am.
job.
So the current occupants in the White House are not known for their forward-thinking nature. And I'm just curious if there have been, in their short-sightedness, any threats to this type of research, looking outward into the future, decisions that need to be made today to prevent disasters later on. I know you said USAID was well-funded for this project.
Yes.
Do you have any thoughts that could help us in terms of where we direct our energy and what we do?
I don't want to restate that one. Did you guys hear her? She's
Yes.
suggesting, could you turn that off if you want me to answer this and not bite the hand that feeds me on No, I'm just No. You just know what he watches. Right? This is probably not going to end
This
there.
is enough.
Okay. Thank you. I never get guffaws. It's really, mostly people just look at me like scientists, like, why are you there? And talking about entertaining myself.
There needs to be a scientific comedy show.
Oh.
Star you.
So here's I have a flip answer that I believe to be true, or we could explore more fully. The occupants of the White House are very afraid of germs. So I'm okay. severely impacted because all of the people below that spot are disabled from running our government, and therefore they have, they spend a lot of time thinking about what those people think about. What would they think of this? I can't make that decision. What will they think of that? They might not like that decision. So we get slowed. Also, our government is just not functioning because we're distracted by everything else. So I am in the 5th year of a second 5-year funding cycle. What month is it? February? February. The federal fiscal year begins October 1. Every other year since we started this in 10 years and my whole 25-year career in science where I've been federally funded, we had to, it was almost like legal, receive our funds for the next fiscal year by September 30th to be able to start the work. It's February, I don't have this year's money. We're still working off of last year's money. And it's not because the money hasn't been appropriated, it's in the budget, it's been designated, it's there. They just don't have any people to process the money to put it into a contract to me.
Mine's a really practical question.
Practical question from
and somewhat selfish. Okay, what I'd like to know is what is the potential for pandemics as a result of travel via airplanes, via ships, via, every time I get on an airplane, I'm wondering if somebody's walked on with some potential pandemic.
Okay, so the potential for spread, which makes something that was just a disease or maybe a local epidemic into a pandemic, is hugely impacted by travel. Okay, we are seeing outbreaks and epidemics on cruise ships. Anybody been on a cruise ship? Norovirus, don't do it. So living closely, just like those pigs and chickens, you push us together closely in tight spaces, even luxurious ones. It's a recipe for disaster. So and I'm not being jokey. It just is. So if you live in a slum tightly together with less opportunity to be apart in air and water and all of those things, we put ourselves at risk. So for amplification and spread, kind of like my other answer, certainly a big issue. Is travel causing epidemics and pandemics? I would say historically, yes, right? We know many of us are of European descent. We came here and killed a lot of people. Our people did. We didn't do it. It's not your fault. But we brought nasty things. So travel can still do that, but the way that we travel is not, we're pretty homogenous now. I can tell you we model that. The computer models, we model that all the time, and we know that things that have what we call a short incubation period, they can infect us and then cause disease fairly rapidly, can get from almost any spot in the earth in 3 days. So if it's somewhere, it's going to be next to you tomorrow, and that's why also we should care, even if we're very conservative about how we spend our money, and we shouldn't be spending money in the far reaches of the earth. We are one globe. We are moving around. So from an infectious disease perspective, we're one big population.
One more question, I think, because
Want to wrestle? Okay.
Me? Okay. A real quickie. What's the relationship of a virus and a prion? It's my understanding a prion is a tiny weeny virus.
No, it's a completely different kind of organism. Yeah. Yeah.
Thank you.
Okay, can I, because it was so quick. Yeah. No, it's its own thing.
I have wildlife.
Wildlife around you. Yes, that's a good thing. Don't be scared.
Rats over my door.
Yep.
And rats and mice.
Yep.
So do I need to really worry about the guano?
Yeah, but don't you anyway from like ick. So this is where human evolution is helpful, right? It's true. One of the biggest things for behavior change is disgust. It's a motivator. Like you, bats around my house. If you can, and I know it's a big if you can make more attractive places, I can give you, I can show you how to make a bat condo. You're not going to do that, obviously. We'll go with your,
My house is architecturally designed for neighborhoods.
They love it, yeah.
And I have a friend who's a bat expert.
Okay. So there are some ways to do that. They aren't always going to attractively please you. So there are lots of things we do with folks all over the world where they don't have doors and windows, so that bats are really loving being in their houses. But there are things like steel wool and chicken wire and things that you can paint to be the same color as your eaves. It's not a great option. The best option, if you can get them to, is you can attract them to a tree in your yard, but not all bats will go to a bat house that you hang up, so you have to get really creative about that. So it's not great. Rodents, you don't really want rodents in your house for all sorts of reasons. And so you'll have to decide. Most of the rodents that are coming in your house are probably not native rodents, they're invasive species. Yeah, in the garage, right? So, I mean, I'm a wildlife conservationist. I trap rats out of my garage. I don't use toxins, but yeah. Because I want to, we need to learn how to live together with the population, so it's more about urban planning, more about where we're going, leaving them places to go, but we bring with us those, Norwegian rats got here because of us. So.
This is the last question over here, one more.
Yes, ma'am.
Thank you. I'm not a biologist, so this may be a naive question, but I wondered whether the logic and methodology that you've been describing and applying to viruses would also apply to bacteria.
Yes, and back to your point, I didn't go there because it will take me, I'll try to be quick, but antimicrobial resistance is a huge emerging infectious disease problem, huge. Dr. Haney here, Krista will tell you about STDs and measles and all the things. It all has to do with how we live our lives, right, and what we choose to and not to do. So antimicrobial resistance and bacteria, huge issue. I don't spend a huge amount of time on bacteria in general because they've been really well studied. So over time, they've caused a lot of disease. They cause epidemics more than pandemics, but they cause a lot of disease. So they've been studied a lot. They're easier to study than viruses, so they cause lesions that can be seen, other than fever, you die kind of things that viruses do. They cause big, pussy boils and stuff, and then people study them. So they've been studied over history a lot more, but we do use our One Health approach to watch for new viruses and antimicrobial resistance. We do the antimicrobial resistance in a little bit different way, with more looking for the genes and not the bacteria themselves.